Hello everyone, welcome back to another session of regeneration biology. So this time we will start with a new model, that is Planaria. Many of you would have heard about Planaria right from your school days. Planaria is a very interesting invertebrate, comes under the group flatworm, and it has got numerous regenerative capabilities. So Planaria regeneration is a very old information for mankind, as old as Hydra. So interestingly, the Planarian regeneration has caught the attention of so many decades or centuries to a differing degree. And different investigators, many of you would have heard about Michael Faraday, who is the father of electricity and who is known for his electrochemical research. He was interested in Planaria research and he has published a paper in 1833. And Morgan, the father of all this genetics and the mutational studies, T.H. Morgan, and he also has contributed significantly to the studies of Planaria using its regeneration. All you need is a blade, a magnifying glass, and the imagination to do an experiment. That's all it was needed, raw material, nothing more. So these are all the papers that is cited here, published. You can see the date, 1833 and 1898. You can imagine how old those research are. A few directives we need to know about the Planaria regeneration. Planaria are blessed with presence of numerous pluripotent stem cells known as C neoblast. Okay. C basically stands for clonogenic, that is the terminology, C neoblast. And that is the name of those stem cell. And roughly around 30% of the Planarian cell mass is these neoblast cells. And there is another category called fate specified stem cell. They are specialized in neoblast, C neoblast and specialized neoblast that provide cellular basis for regeneration in Planaria. Positional information that is harbored in the muscle, and that is another information that is you need to know, that Planaria, if it lost a part of its body, it has to be restored back purely because of the positional information. Positional information in a strict sense, you can say, you know where your nose is, where your eye is, where your mouth is, or where your hands are there. These are all nothing but positional information. You will not see a hand coming out of your forehead. You will not see a nose coming out of your chest, because these are all positional identity genes are established right from the embryonic development. That is why eyes are in the place of eye, head is in the place of head, tail is in the place of tail. Okay. So this information you should keep with you, that the information that is gathered and located in positions, different positions, determine what part is formed. How the combination of positional information and its influence on this neoblast contribute to the regeneration. This we will study. And how progenitor targeting by extrinsic cues and self-organization that can combine to determine where the regenerative progenitors go. So these are all things which we need to address in the future classes. So in a simplistic sense, you can see this is a picture, it's a cartoon of Planaria. You can see different types of amputations can be performed. What you see in this blue colored animal is, it is irradiated. What you don't see blue color, as you can see here, this dotted, it is normal. So in a normal Planaria, you chopped off the head and you can see that head is regenerated after some time. Whereas in an irradiated Planaria, you chop off the head, new head doesn't form, and eventually the leftover part of the body will disintegrate. That is what has been shown here. And now you take the Planaria into two halves, anterior half and posterior half. Anterior half you irradiate it nicely, and then you chop off its head. Anterior half has the head also, okay, and then you chop off its head. Slowly the head will regenerate back, as you can see here. So it tells you something. If the posterior half has normal good cells, no radiation has been given. Radiation, all of you know, it will cause DNA damage and cell death. So posterior part did not receive, the backward part did not receive any radiation, hence the head could regenerate. That means something from the posterior healthy part moved into the dead part. Now comes the very interesting experiment where a normal planaria and a radiation killed planaria. What you did, that radiation killed planaria, it do not regenerate, I have seen it, and it will eventually die. Into the radiation killed planaria, you took a small tissue piece from a donor planaria which is normal, and you implanted it, and then you chopped off the head. Then it can restore the head, and also it will restore the normal body. What it tells you, that something from this donor planaria, which is nothing but did not receive a radiation, that is the only difference, it could contribute something. So you can see here, results in complete regeneration. When you normal situation, it results in complete regeneration. But if you have a heavy radiation, and not only the head will not regenerate, but it will disintegrate also. And on the other hand, that can be rescued by putting a small tissue from a donor. And this is something very interesting, that this is experiment done by Wolf and Lender in 1962, and we learned something that is coming from a healthy planaria is able to restore normalcy. Now, another interesting feature about the planaria is you can cut between the eyes of the planaria. You can see planaria has got two eyes, as you can see here, depending upon which species you are talking about. You just cut between, start from the tip of the head until the middle of the two eyes. All you need to do is just to cut it. You will end up getting two-headed planaria. It will not become two planaria, it will become two-headed planaria. Again, you cut it, you will get four-headed. And like that, you can get 1, 2, 3, 4, 5, 6, 7, 8, 9, 10-headed planaria, you could see here. So you can keep cutting, it will keep making more and more and more of two heads. But the planaria remains the same. Again, you chop off the body into a small slice, very small slice, took from somewhere in the belly region. You end up having two heads on either side. The actual head region also you will get a head, and the posterior region also you will get a head. So double-headed planaria can be obtained. And also, planaria can do some very unique way of reproduction, similar to that of what bacteria will do. Bacteria will reproduce by binary fission, right? So planaria can reproduce by something called paratomy. Paratomy means, and this is been shown in Planaria fusipara. Different planaria has been used for various experiments. You will see different species in a while. Before the fission at the level indicated by the arrow, the posterior part of the planarian has already begun to transform into a new individual. As you can see here, it was a proper planaria, then it started developing a fission, just like how binary fission happened in bacteria. And then it started developing two eyes, etc. Finally, this will pinch off and you will end up getting a new planaria. So, planaria uses stem cell mediated regeneration approach. You can see here, this is a way of asexual reproduction where a planaria can be split into two, just like how if someone gives you a stick, you can start longitudinally, cut it, and it will become two pieces. Same way, the planaria split into two, and each half will give rise to the last part, and that will become two planaria. Planarium flatworms reproduce by binary fission. So previous picture you saw, it is cutting left, top and bottom, or anterior and posterior. Here you are cutting left and right. So splitting the body from top to bottom and regenerating the right and left halves. The cells capable of doing this pluripotent are called the pluripotent stem cells, known as neoblast, and they are used for the repair and also for the regeneration of the lost part. So from 1700, it is known, year 1700, it is known that the planarians are cut in half and it can give rise to multiple pieces. It's one of the ancient knowledge. Same way, T.H. Morgan also T.H. Morgan and Child, another scientist, they realized that such polarity indicated an important principle in the development. Polarity means what? Head region has got head, tail region has got tail. This is polarity. You also have polarity, your body. You don't have head in your base of your body, or near your leg you don't have head. Your head is in the place where head should be there. Your legs are in the place where leg is there. That is polarity. So Morgan pointed out, when a head and tail were both cut off, the middle piece would always regenerate, the head from the former anterior region and a tail from the former posterior region. It doesn't happen other way around. and never the reverse. If, however, the middle piece was too small, then the regenerating portions would be abnormal if it is too small. Based on these observations, they postulated that a gradient of anterior producing material emanated from the head. The middle segment would be told what to generate, and then two ends by concentration gradient of these two materials. You can see here, this is cut into two pieces. The anterior piece made the posterior piece, and the posterior piece made the missing anterior piece. And you cut into three pieces, you end up getting regeneration from, this is understood, the middle portion made a new planaria. But if you make a small piece, you end up getting a head from both the sides. So if the piece is so small that the gradient is not worthy enough or powerful enough to distinguish which is anterior, which is posterior, you end up getting two heads. And you can see there are so many examples of planarian species are there. You can see, no need to go through it, but the morphology shows their names are listed here, and different morphologies are there, and some planaria do not have the ability to regenerate. Don't think every planary can regenerate. The planarian species, some of them do not have the ability to regenerate. We'll see them in a short while because this is a introductory class. A few more facts we need to know about the planaria. Planaria can grow and literally degrow. That means shrinking. It can shrink back by the food supply availability in the environment, dependent on the adjustment of an organismal turnover rates. Scaling the body plan proportions over as much as 50-fold. That means a planaria which is 50 kilo in size, I'm giving an arbitrary number, no planaria is 50 kilo. It can go as small as 1 kilo. That is 50-fold shrinking is allowed based on the availability, and it can bounce back just like human liver. We discussed about liver, right? Their dynamic body architecture further allows astonishing regenerative abilities, including the regeneration of complete and preferably proportioned animals, even from tiny tissue remnants. If you have a small tissue, it will assume a shape of a small planaria and then it grows back. Scientists use two species, that is Smithia mediterranea. In short form, it is called as SMED. Planaria papers, you will see SMED, S-M-E-D. That means it is for Smithia mediterranea. And another species is Dugesia japonica. In short form, it is called as DJ. Because both have excellent regenerative capabilities, and clonal strains originating from single animals are used in labs. They are clones. Because to make the clone, it's very easy, just like animal tissue culture. Take a planaria, cut into 3 pieces, you end up getting 3. You want more planaria, cut into 10 pieces. You will get 10 animals. So it's like a meat piece, keep growing. So you don't have any difficulty to maintain them in a clonal manner. Vegetative propagation is very easy. Planaria has an abundance of adult stem cells known as neoblast. They are collectively referred to as neoblast. Transplantation of a single neoblast into a lethally radiated, like in the previous slide we saw, take a planaria and irradiate thoroughly, you end up getting disorganized or completely damaged planaria. It will not grow, it will not survive. But into that planaria, irradiated planaria, you transplant one cell from a healthy donor. The entire animal will come back, but that will be clone of this donor. Somewhat like you may have heard about how animal cloning is done. They take the nucleus from a donor and put it in the recipient zygote. You end up getting clone of that nucleus owner, something like that. And in a stem cell depleted irradiated planaria, you can put one neoblast from a donor. It will, the recipient, give rise to perfectly healthy animal of the donor genotype. Donor genotype means whose C neoblast you took it. This paper was published in Cell Stem Cell a few years ago, suggesting its totipotency status. Totipotency means what? If you can make one animal from one cell, that is called a totipotency. However, understand, if you take a C neoblast and put it in a medium, it will not give rise to an animal. So what it tells you, C neoblasts, although they are totipotent, they need the guidance clue from the extracellular matrix or the positional information. Because of irradiation, nothing will happen to the proteins. They will stay there and nothing will happen to the matrix. DNA gets damaged and cell eventually will die. So this is what you should keep in your mind. A further unusual feature of Planaria neoblast is their high basal mitotic activity. They keep growing, keep dividing. So a Planaria tissue keep on replacing. If Planaria has got an eye, last month is a different eye, this month is a different eye. It is completely revamped. It's just like, you keep buying furniture for your house. Your furniture is staying only 1 month in your house. You threw it and bought a new furniture. Like that, Planaria keep doing. Their cells constantly keep on. So there is none of its organs are old. Every time it is new. From credit goes to the C neoblast. Remember, this happens in a steady state level. No regeneration is needed. It constantly regenerates every organ. So cellular and molecular basis of Planaria regeneration, if you look closely, Planaria regeneration, it involves the production of something called blastem. Like in liver, you don't see it. In Hydra, you don't see it. But in Planaria, you see the formation of a group of cells. They are disorganized. They don't have any tissue specificity. And that is called a blastema cell. We will see about blastema more in the future classes. Because a small Planarian body fragment cannot eat until suitable anatomy has been regenerated, including pharynx and brain. What if the broken fragment of Planaria doesn't have mouth? It cannot eat, right? So there is no food supply. So until the mouth is developed, it has to continue to survive on the available resources in the form of glycogen, proteins, lipids, etc. in its body. So missing tissues thus cannot be regrown at their original scale. Missing tissues cannot be regrown at their original scale. Thus, it cannot be regrown at its original scale, but it will form in a miniature form. The blastema thus typically only regenerates some of the missing tissues, such as head to start with. Say, if a body is cut, the anterior piece and posterior piece is having only half of the Planaria body, it will not create the rest of the anterior portion. It will just create a head right in the tip of that posterior portion, and then it will keep growing, building the bulk. This is the way in which they grow. And some tissues will initially be overabundant in this case of repurposing and in the amputated fragment. So sometimes when this repurposing is going on, the amputated fragment will have somewhat more than adequate or more than required tissue will be there, so they will undergo apoptosis, and sometimes a required tissue will be missing. Then the existing tissue will take over the function of that tissue, and it will keep changing slightly for time being, but it's okay simply because it has got C neoblast, and it will keep revamping it, and the proper cell will be replaced back. And its position and relative position of the organs has to be maintained, and it will be taken care of by the position control proteins, which we will see in the future classes. The changes that is leading to mini Planaria formation have been referred to as morphallaxis, because it is done by repurposing, like we saw in the case of Hydra. Morphallaxis is largely the result of new cell production and cell loss, rather than changes in the differentiated state of the cell. So they are, like I told you, they are repurposing, and that is forming a miniature Planaria, because the miniature Planaria, if it has a mouth that is developed because of the repurposing of the tissue, then it can start eating. So this is the overall idea of restoring a miniature Planaria to start with. So Planaria can also regenerate missing tissues on their mediolateral angle. Mediolateral angle means, like you can say, you have a chest, you have a sternum right in the center of your body, and to your right hand, that is mediolateral angle. So your chest to your right hand is one mediolateral angle, and your chest to your left hand is another mediolateral angle. So it can make organs that side also, not that it has to be cut horizontally or longitudinally. And means it can have sagittal and parasagittal section. So partial amputations can result in duplicated structures, like we saw in the 10-headed Planaria. Partially you are cutting it, you end up getting 2 heads. A sagittal incision between the eyes, for instance, can result in the regeneration of 2 heads, and you can keep repeating it. But there is something interesting that will be coming, we will discuss. Removal of irregularly shaped tissue fragments. Say it end up getting 10 heads. Now what you do, you did one amputation and got rid of all the 10 heads. Can you get 10 heads back? The answer is no, because the animal's blueprint says it has to have only 1 head. So you ended up making 10 heads. You cut with one cutting, you got rid of all the 10 heads. you end up getting only one head back. So irregular shaped tissue fragments can result in regenerative intercalation of the missing anatomy. That is called intercalary regeneration. That means that regeneration happened because of the way of living, a time forward. It is trying to adjust to the stress, and that results in regeneration. Sometimes you can make multiple eyes also just by amputating eye alone. That also we will see in the coming classes. But what I'm saying is you can create an abnormal development or regeneration in planaria. But once you get rid of all the abnormal region, the normalcy comes back. Between the fused wound phases, you can end up getting complicated structures. Similarly, one region of the body can be transplanted next to another and regenerative responses can be triggered, including the formation of outgrowths that appear to intercalate missing coordinates between juxtaposed tissues. Some pictures we have so that you will know, it's easy to understand. In this picture, you have seen it is cut, head is cut, tail is cut. So naturally, from the piece fragment 1, the rest of the body is formed. The brown color is the actual, and the ash color is the newly formed part. From the middle, from the side part, from the posterior part, the remaining part will be the fragment 2. The head is formed back. But most interesting thing is this third piece where it is taken a small piece. And you can see here from the small piece, it got head and left side of the body and also the bottom. So this is something very interesting. And you can see what is mentioned blastema here, that is the disorganized tissue that is formed immediately. And it's panel B, and regeneration from a fragment that results in a small animal that can eat but grow towards the original size. You can see a small fragment and it gave rise to a miniature organism, and a big fragment, it also gave rise to a miniature organism. Eventually both of them will give rise to a proper organism. This is the most interesting part. Here, if you cut it, one small piece is removed and then you have a big brown piece. So the big brown piece made the missing small piece, whereas this missing small piece made an entire normal organism, proper looking organism. It did not even had pharynx, you should remember. So the center portion is pharynx, eating, and mouth is not here. Head and eyes are here, but pharynx is in the middle. Same way you cut into 2, you end up getting 2-headed. If you cut again here, you will end up getting 4-headed. Both the sides you cut it. Now, another exciting part is you have an anterior portion and a posterior portion, and you got rid of this pharynx part. Then it will selectively, and you fused it. You took this anterior portion and the posterior portion and you fused it together. This middle portion you removed it, but you will end up getting this middle portion grown back. So in this experiment, what they have done is they took this head piece and this tail piece and fused. This much portion is been thrown, kicked out, but that portion grows back. So the blueprint of the organism knows which portion to restore back. So initially it will do the tissue rearrangement and do the morphallaxis, and that is further favored by blastema formation and the growth of the proper head or tail or pharynx, whichever part is missing. So the clonogenic neoblast, they are basically the pluripotent stem cells that produce the missing cell types. So the Planarian regeneration that can involve new tissue production in the blastema. So the blastema always will have new tissue that will be produced, and that has to now differentiate as expected or as required, because a small planarian body fragment cannot eat until suitable anatomy has been regenerated, including pharynx and brain. Regeneration must occur with existing resources, otherwise it will not happen. Blastema formation typically only regenerate some of the missing tissues such as head, and is coupled with changes in the pre-existing body regions for the regeneration of other missing tissues. So I will explain once again. The blastema formation typically only regenerates some of the missing tissue. Although blastema has got all tissues capable of producing all cells of the organism, it will not make all tissues. In the head region, it will make from that blastema, although it is a totipotent cell mass, it will make only the head out of it. Is coupled with the changes in the pre-existing body regions. It will rearrange so that you will end up getting only the missing part. So these changes have been referred to as the morphallaxis. So the missing tissue also can respond, and by proliferative and cell death response. So neoblasts respond to the injuries by increasing their rate of proliferation. So whenever there is a amputation, say let us assume a given planaria has got 1000 neoblasts and they are dividing once every day, let us assume. So if there is an amputation, now these 1000 neoblasts, of course 1000 would have come down because neoblasts are distributed throughout the body. They don't have any reservoir. So if a 30% of the body is cut, this 1000 neoblasts will now come down to around 600 plus, right? So instead of dividing once in a day, they will divide once in every 1 hour. So its a rate of proliferation increases. So the body see neoblast, know whether there is an amputation occurred or not. So the neoblasts respond to injuries by increasing their rate of proliferation. And that's not it. It should reduce the apoptosis rate also. An initial peak of proliferative response is widespread, occurs approximately 6 hours after injury, and it is followed by second phase of sustained proliferation near the wound, occurring by approximately 48 hours after injury. So within 6 hours, it will respond by adequate proliferation and is followed by a second phase of sustained proliferation near the wound site, which leads to the production of blastema. Blastema happens only in the wound site, and this happens around 48 hours. This increased proliferation that is occurring throughout the body, now they all will kind of filter out, just like, they all will move towards the tip and they will form a mass lump of cells that is called blastema by around 48 hours. The initial proliferative response occurs after any injury type. Any injury. Injury can be amputation or removal of one eye or a removal of pharynx, etc., including injuries such as incisions that do not require substantial production of new tissues. Sometimes when you are injuring, like you did a cutting, you cut across. So, but since you cut across 2 eyes, it knows it will make both the cut will make the missing part. That is why you end up getting 2-headed animals. So here the logic of the animal, maybe I will go back to that picture once again, then after completing this. By contrast, the second phase of proliferation only occurs at wounds that can result in the substantial tissue loss and is referred to as missing tissue or regenerative response. Keep this word in mind, missing tissue response or regenerative response. They are slightly different, and this response is associated with neoblast accumulation at the wound, which leads to the blastema. Let us see this picture once again. If you cut the animal into 2, as you can see here in this panel D, if you cut into 2, the immediate response of this fragment, like you saw here, this piece, this tiny piece, this tiny piece was trying to make the entire organism like that. This is cut only up to here. You did not chop it off. This piece, immediate goal is to make the remaining missing piece. It do not know it is there in the neighborhood. It will not try to fuse it back. So this left hand piece, it made a proper head, and the right hand piece made a proper head. This can be continued for a prolonged amount of time. That is what you see, that the morphallaxis make sure a rearrangement of these cells. However, in this kind of cutting where you did not bring about a morphallaxis response because it has got the head, proper head is there, you did not remove the head, but that local disruption was powerful enough to replace the missing part. So in this way, we kind of confused the animal whether it's not a proper amputation. Actually, there was only a wound healing was needed, but the animal is so plastic, or neoblasts are so determined, they made a proper head. So that is what we should understand here. The response, by contrast, the proliferation can occur specifically in the missing part. This will eventually lead to a proper formation of the structure when there is an amputation. Otherwise, it will simply make the missing part. We'll continue to learn about planaria regeneration in the next class. Thank you.